Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start a topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with virus addition by glycoproteins. In today's session, we will start with catabolism of glycoproteins. Then we will have all diseases due to carbohydrate metabolism. Lastly, we will start with a new chapter, digestion of fatty acids. Good morning everyone. Let us start a topic. The catabolism is by hydrolysis, which are bond specific. Deficiency of these enzymes would result in marked accumulation of substrates. The half-life of many proteins in circulation is determined by glycosylation. This is a potential area of therapeutics where carbohydrate side chains are removed, that is asialoglycation, and the half-life of the proteins used as drugs can be prolonged. These group of inborn errors of metabolism characterized by excessive intralysosomal accumulation of glycosaminoglycans in various tissues. They are progressive disorders. The clinical manifestations include coarse facial features, thick skin, and corneal opacity due to accumulation of glycosaminoglycans. Mental retardation, growth deficiency, skeletal dysplasia are also seen due to defective formation of ground substances. In general, the effective degradation of heparan sulfate leads to mental retardation predominantly, whereas accumulation of other glycosaminoglycans leads to mesenchymal abnormalities. A characteristic finding in this is the excessive excretion of any one of the glycosaminoglycans in urine. All these diseases are inherited as autosomal recessive traits except Hunter's disease, which is X-linked. This is a diagram showing on the left side dysfunction gag layer, which includes bacteria and urine solutes, whereas on the right side there is normal GAG layer, which includes proteoglycan and water. Coming to mucopolysaccharidosis. Type 1, also known as Hurler's disease. It occurs due to deficiency of L-iduronidase. Mental retardation, skeletal deformity, corneal opacity are present. Dermatan sulfate and heparan sulfate in urine are seen. Then type 2, which is also known as Hunter's disease. It is due to deficiency of iduronate sulfatase. Mental retardation, skeletal deformity, and deafness are seen. No corneal clouding is seen, but dermatan sulfate and heparan sulfate are seen. Type 3, also known as Sanfilippo's disease. Occurs due to deficiency of N-acetyl glucosaminidase and heparan sulfatase. Mental retardation, skeletal deformity, corneal clouding are seen. Heparan sulfate is seen in urine. There are 3 different types of diseases reported. Type 4, which is also known as Merck's disease. It occurs due to deficiency of galactosamine sulfatase and beta-D-galactosidase. Its mental retardation, skeletal deformity, epiphyseal dysplasia, and corneal opacity are seen. Keratan sulfate and chondroitin sulfate is in urine is seen. There are 2 types of diseases reported. Type 5, Shy syndrome. It occurs due to deficiency of L-iduronidase. Here no mental retardation is seen, but mild skeletal changes and corneal opacity is seen. Dermatan sulfate is seen in urine. Type 6, Marotox Lammy's disease. Occurs due to deficiency of N-acetyl beta-D galactosamine 4 sulfatase. Skeletal deformities and corneal opacity are seen. No mental retardation is seen, but dermatan sulfate is seen in urine. Type 7. Slice disease. It occurs due to beta glucuronidase. Mental retardation is seen. Dermatan sulfate and heparan sulfate are seen in urine. Coming to other errors in carbohydrate metabolism. Glycogen storage disease. Type 1, which is also known as von Jurkes disease. It occurs 1 in 1 lakh births. It occurs due to deficiency of glucose 6 phosphate and it is located at chromosome 17. Hepatomegaly, cirrhosis, hypoglycemia, ketosis, and hyperuricemia are seen. This is a diagram showing von Jurkes disease. Here in the brain, the hypoglycemia occurs, which leads to seizures, coma, and intellectual disability. Then glycogen is stored, hence kidney enlargement is seen. Then hepatomegaly is seen, which leads to distended abdomen, and doll-like faces is seen. Type 2, Pompe's disease. It occurs 1 in 1.75 births. It occurs due to deficiency of lysosomal maltase and it is located at chromosome 17. Generalized glycogen deposit and lysosomal storage disease are seen. Type 3, Cory's disease. It 1 in 1 lakh 25,000 births. It occurs due to deficiency of debranching enzyme and it is located at chromosome 1. Hepatomegaly and cirrhosis are seen. Type 5, Anderson's disease. It occurs 1 in 1 million births. It occurs due to deficiency of branching enzyme and it is located at chromosome 3. Hepatomegaly and cirrhosis are seen. Type 5, Mucopolysaccharidoses disease. Occurs in 1 in 1 million births. It occurs due to deficiency of muscle phosphorylase and it is located at chromosome 11. Exercise intolerance is seen. Type 6, Hirsch's disease. It occurs in 1 in 1 million births. It occurs due to deficiency of liver phosphorylase and it is located at chromosome 14. Hepatomegaly and hypoglycemia are seen. Type 7, Thyroid's disease. It occurs in 1 million births. It occurs due to deficiency of phosphofructokinase and it is located at chromosome 1. Type 8, 1 in 1 lakh 25,000 births. It occurs due to deficiency of phosphorylase kinase and it is located at chromosome X. Fructose intolerance. It occurs in 1 in 120,000 births. It occurs due to deficiency of aldolase B and it is located at chromosome 9. Hypoglycemia, vomiting, and hepatomegaly are seen. Fructosuria. It occurs in 1 in 1.3 lakh births. It occurs due to deficiency of fructokinase, but it is a benign disease and urine sugar is seen. Galactosemia. It occurs in 1 in 35,000 births. It occurs due to deficiency of galactose 1-phosphate uryl transferase and it is located at chromosome 9. Hypoglycemia, hepatomegaly, mental retardation, jaundice and congenital cataract are seen. A variant of galactosemia, 1 in 40,000 births. It occurs due to deficiency of galactokinase and it is located at chromosome 17. It shows congenital cataract. essential pentosuria. It occurs in 1 to 2500 births. It occurs due to deficiency of xylitol dehydrogenase. It is a benign disease. Pyruvate carboxylase deficiency. It occurs in 1 in 25,000 births. It occurs due to deficiency of pyruvate carboxylase located at chromosome 11. Mental retardation is seen. Glucose 6-phosphate dehydrogenase deficiency. It occurs in 1 in 5,000 births. It occurs due to deficiency of glucose 6-phosphate and dehydrogenase, and it is located at chromosome X. Drug-induced hemolytic anemia is seen. In this diagram, we can see glucose 6-phosphate is converted into 6-phosphogluconate in the presence of glucose 6-phosphate dehydrogenase, which is inhibited in the previous slide. Hexokinase deficiency. It occurs due to deficiency of hexokinase, and it leads to hemolytic anemia. Pyruvate kinase deficiency. It occurs due to deficiency of pyruvate kinase, and it also gives hemolytic anemia. Pyruvate dehydrogenase deficiency. It occurs in 1 in 2.5 lakh births. It occurs due to deficiency of pyruvate dehydrogenase, and neuronal loss in brain, muscular hypotonia, and lactic acidosis are seen. Coming to metabolism of fatty acids. Digestion of lipids. The major dietary lipids are triacylglycerol, cholesterol, and phospholipids. The average normal Indian diet contains about 20 to 30 gram of lipids per day, whereas Western diet generally contains 2 to 3 times more the quantity than this. Coming to digestion in stomach. The lingual lipase from the mouth enters the stomach along with the food. It has an optimum pH of 2.5 to 5. The enzyme therefore continues to be active in the stomach. It acts on short chain triglycerides. The short chain triglycerides are present in milk, butter, and ghee. The action of lingual lipase is observed to be more significant in the newborn infants. Gastric lipase is acid stable with an optimum pH around 5.4. It is secreted by chief cells. The secretion is stimulated by gastrin, and up to 30% of triglycerides occurs in stomach. Coming to digestion in intestines. Emulsification is a prerequisite for digestion of lipids. The lipids are dispersed into smaller droplets, then surface tension is reduced and surface area of droplets is increased. The process is favored by bile salts, which brings about detergent action, peristalsis, which brings about mechanical mixing, and phospholipids. Here ends the session. Kindly like, share, and subscribe our channel. Thank you.